This is Unit 8, Part 3. The pyloric valve, or the pyloric sphincter, same thing, which demarcates the boundary between the stomach and the small intestine. Okay? So, the small intestine runs from the pyloric sphincter, or pyloric valve, down to the ileocecal valve. The ileocecal valve is at the junction between the ileum and the cecum. That's why it's called ileocecal. You see that right here, ileocecal. That's the junction between the ileum and the cecum. So it goes down to the ileocecal valve. There are three sections, subdivisions anatomically, within the space or length of the small intestine. The first is the duodenum, and after that comes the jejunum, and then the ileum. There are ducts which drain secretions from the liver and from the pancreas into the duodenum. They are the bile duct and the main pancreatic duct, and they drain into the duodenum. The bile duct delivers bile, and the pancreatic duct delivers pancreatic enzymes. The distal part of the small intestine, the ileum, joins the large intestine at the ileocecal valve. Okay, now here, this is a diagram that just shows the bile duct, okay? And it also shows the main pancreatic duct. There is an accessory pancreatic duct, but there is also the main pancreatic duct and the bile duct. The bile duct leads down from the liver and from the gallbladder. The gallbladder acts as a reservoir for bile. You don't always need bile to be sent into the duodenum. This section here, where these ducts are entering into, this whole section here is the duodenum. Okay, so products from the liver are sent down the ducts coming from the liver, and they get stored in the gallbladder, and when they're needed, the gallbladder contracts and squeezes secretions down the bile duct into the duodenum. And the same goes for the pancreatic enzymes that are secreted by the pancreas for digestion. Those also go into the duodenum when needed. That's highly regulated, all of that. In terms of microscopic, smaller level anatomy, or well, not macro, but at a more closer level, there are a lot of modifications of the wall of the small intestine, all of which act to increase the surface area, because the small intestine is not only involved in as a site for hydrolytic enzyme activity and breakdown of chyme coming from the stomach, the material which was food, gets broken down, but it also is where a lot of the absorption occurs of individual building blocks that have been produced from those proteins, carbohydrates, lipids, and nucleic acids. For all of those building blocks which are now ready for absorption, to be absorbed. So they get absorbed across the wall of the small intestine. A lot of that's occurring in the duodenum. And there are many modifications to the small intestine wall in order to increase its ability to absorb. Though all of those modifications, what they do is they increase the surface area. The larger the surface area, the better the absorption. The modifications include the plicae circularis, which are these large folds, deep circular folds of the mucosa and submucosa. There are the villi, which I've talked to you about before, which are finger-like extensions coming out of the mucosa. They're very tiny. And at the very top of each individual columnar epithelial cell in the simple columnar epithelium that lines the gut, the simple epithelium making up that innermost surface of the mucosa, there are tiny projections extending from each cell, and they're called microvilli. And they look as follows. Here you see increasing magnification. Here's the entire duodenum. Then they're magnifying just a small area. This small square is magnified in this diagram. And then this is the same as the middle diagram. Oh no, wait. And then this is a further magnification so that you have just this small area here being magnified in this diagram, and they're actually just showing one cell and just a bit of the next cell over. This, of course, is a photomicrograph taken with a microscope to show you the villi in actual the actual appearance of the villi. So what we have here, where we are looking at the entire inner surface of the small intestine, is these folds. Along here we have these folds. The surface is folded back and forth, so you get these ridges, these circular ridges or folds, and those are called the the plicae. Okay. At the same time, if we look closer at a higher magnification, you can see that there are also small finger-like extensions at the wall, and those are magnified here in this diagram. And you see these finger-like extensions. This is the villi, and you have the mucosa. The mucosa, of course, is made up of the epithelium, a simple columnar epithelium that covers the surface. I'm going to change the color. This is the simple columnar epithelium. Simple meaning one cell layer thick, and columnar is the shape of the cells, and they cover, that's in cross section. So all of this pink stuff here, all of that pink, all those villi, they're all covered with a single layer thick, a layer of single cell thick of epithelial cells, and beneath that is the lamina propria, and inside the lamina propria you have, as shown here, you have these blood vessels, and you have it's a specialized part of the it'll come to me in a second. It's a specialized type of lymphatic vessel which drains tissue fluid. It's called a lacteal. So down the center in green, shown in green, down the center of each villus is a lacteal, and that's involved in absorption of products of digested lipid. So fat gets absorbed into that specialized type of lymphatic vessel. The blood capillaries are involved in absorption of breakdown products from protein, carbohydrate, and nucleic acids. In the simple columnar epithelium that lines the surface of all these villi are cells that produce a protein called mucin, and when mucin is secreted and comes into contact with water, it becomes mucus, and those cells are called goblet cells. These so-called absorptive cells are in fact the simple columnar epithelial cells that allow absorption, and they are highlighted further here in this further magnified diagram, which shows the cells, the simple columnar epithelium, and the fact that they have at their very top surface very tiny microvilli. Do not confuse villi and microvilli. Villi refers to each of these finger-like projections that stick out from the side wall of the intestine. And microvilli refers to the small projections that are actually at the surface of each and every cell. Okay, those are microvilli. Here in this photomicrograph, you see very clearly the simple columnar epithelial layer going all over the surface of this villus, and here's another villus beside it, part of another villus beside it. And if you look carefully, you can also see some goblet cells. There are goblet cells all along in different spots. I'll put a dot on each. There's a goblet cell there. There's a goblet cell there. There's another one there. Here, there's two. There's one. There's one. Okay, there's one. So there are goblet cells all along, sprinkled all through that simple columnar epithelial layer. Let me just get rid of these circles. Okay, and of course you have the interior lamina propria, and although you can't see it well, all through here, that's all lamina propria inside the villus, and although you can't see it, there's a lacteal, and there are blood vessels in there for absorption. Let's move on. The simple columnar epithelium of the mucosa is made up of the absorptive epithelial cells. These are columnar epithelial cells. There are goblet cells producing mucin, which will become mucus. There are some enteroendocrine cells, cells that produce hormones, and there's a sprinkling of immune cells called T lymphocytes. Okay, the T lymphocytes are sprinkled through that area. In some parts of the small intestine, there are areas of congregated areas of T lymphocytes or B cells, B lymphocytes. There's areas that have a whole bunch of lymphocytes in them, and we'll see them later. The intestine also has what are called crypts. Okay, these are glands in the wall. The epithelium dives down. It was on that diagram, a crypt. It dives down as a duct going down. The epithelium follows down into a duct that goes into the wall, and it's able to, that duct has cells in it that secrete some intestinal enzymes. As I said, there are also accumulations of immune tissue lymphocytes that are found in the wall. This is, of course, in the submucosa, not the mucosa, and they're called Pear's patches. There are some glands also that secrete an alkaline mucus to protect against acids that's coming in from the stomach. This so-called intestinal juice is everything that gets secreted by the glands that line the wall of the intestine. The crypts they're found inside the bottom of the crypts. They're producing their secretions in response to being distended or stretched because food is present, or when they're irritated because there's food present. It'll trigger a reaction, which is secretion of intestinal glands secreting the intestinal juice. It is those secretions are slightly alkaline to try and counteract the acidity of the material coming from the stomach. It's largely water. It's some enzymes in it, not a huge amount, and mucus. Now the liver is a very important accessory organ for the body. You cannot live without a functional liver. People in liver failure need either if their liver is failing and cannot recover, then they either need a liver transplant or will die. It is, in fact, could be considered, it's a gland, it's an exocrine gland, it secretes material out, but a lot of people think it secretes enzymes. It does not. It does not secrete any enzymes at all. Okay, it does not produce enzymes for digestion. It produces other material that is helpful in digestion called bile. It is the largest gland in the body. It is an accessory digestive organ. It has 4 large main lobes. And here you see the liver, and the liver is positioned mostly on the right-hand side. I'll outline it here. You can see liver is, all of this is liver. All of that is liver, mostly on the right-hand side. Mostly under the lower end of the rib cage on the right hand side, but still extending below that. The liver, of course, drains its product, bile, out through the hepatic duct, and that there are a couple of hepatic ducts draining bile out of the liver. These fuse to become the common hepatic duct, and that fuses with what's called the cystic duct, which together they form the bile duct itself. So here you see the right and left hepatic ducts of the liver fusing together to form the common duct, which then fuses with this cystic duct coming from the gallbladder to form the actual bile duct that leads down to the duodenum. Notice that there are sphincters which can control whether secretions come out of the bile duct, and also whether there are and they can control also secretions coming out from the pancreatic duct. So these sphincters positioned here down at the end can control flow from these ducts, flow of bile or pancreatic enzymes can be controlled by those muscular sphincters. Now the anatomy, at a microscopic level, the anatomy of the liver is interesting. I'd say it's the main bulk, or the main part of the tissue of the liver is made up of cells called hepatocytes. That's the proper name for liver cells. You can call them liver cells, but they're hepatocytes. The overall structure that you see throughout the liver are these 6-sided lobules. These hexagonal or 6-sided, hexagonal shaped or 6-sided liver lobules are long columns, okay, that in longitudinal section would look like a very long column. In cross section they are hexagonal shaped, and you can see very clearly these hexagonal shaped liver lobules, but they're actually cross sections of these 6-sided columns. In this photomicrograph you can see that each one, in this case, in this tissue, is surrounded by connective tissue, and it's 6-sided. So that's very clear. I can tell you that this is not human, because the microscopic anatomy of the liver is the same, but does not have this surrounding band of connective tissue. Okay, so that's absent in the human. It's just each liver lobule in a human is adjacent to the next one. So these two would be, in a human, would be touching each other. That band of connective tissue would be absent in a human liver. So I can tell you that this comes from an animal other than some animal other than a human. Okay, in the middle of the lobule you have what's called a central vein. And we're going to look at this in more detail in the microscopic anatomy on the next slide, I believe. No, maybe it's the next one after this. The liver cells have a lot of functions. They produce bile. They process nutrients that are in the blood coming into the liver from the small intestine. They act as a storage, the hepatocytes, the liver itself is mostly hepatocytes, and these cells act as a storage site for fat-soluble vitamins. And they also act to detoxify certain toxins that can be present in things that we eat. The bile that the hepatocytes produce flows between the hepatocytes towards the bile ducts, and I'll explain that to you, I believe, in the next slide. In the liver, there are also cells that are involved in removal of bacteria from the blood, and these are macrophage lineage cells. They are derived from macrophages. Macrophages are phagocytic cells that are white blood cells that are found in the blood, but these are macrophages found in the liver that are not moving around. They don't go into the blood and move around. They are in the liver, and they're called Kupffer cells, and they're important for removal of any bacteria that could be in the blood, the nutrient-rich blood that's coming from the intestine into the liver. All right, so nutrients are picked up from the gut. Food is digested, broken down by hydrolytic enzymes, then absorbed across the simple columnar epithelium into the blood vessels in each of the villi, and then that blood, which is venous blood, flows back, and before it goes back to the heart, it first goes through the liver, where it gets processed in all these ways, and Kupffer cells present in the liver can remove any bacteria that manage to get across the simple columnar epithelium. It does not happen often, but it's such a huge surface area, and there are so many bacteria in the contents of your gut that it does happen on a fairly regular basis, and you have to have some sort of protection against that, and it is the Kupffer cells in the liver that do that. Blood returning from the intestines first goes into the liver, where it gets processed, and in that processing, one of the steps is the Kupffer cells phagocytosing and killing bacteria. So here we see again this diagram showing I've magnified it a bit, I've expanded it. So we see this lobule, cross section of a liver lobule, and you see the central vein where blood is draining towards this central vein, and that's what leads out of the liver, and all that blood goes back up into the leaves the liver after it's processed, and it leaves the liver following processing. I'm going to make it red to show it's blood, and goes back to the heart. Okay, it's a little more than a little difficult to see, but you can actually see individual hepatocytes here. They are pretty small, but you can see individual nuclei. I think inside there's one hepatocyte with a nucleus in it. There's another one. There's another one. Okay, they're about that size. So you can see that the blood is interacting with those hepatocytes and being processed. How does that happen? Well, blood coming from venous blood that drains the intestine, that is filled with nutrients if you had a meal and were digesting your meal, comes back, the venous blood comes back into the liver, and it enters into the liver through what is called the hepatic portal vein. So, and the hepatic portal vein, branches off into many tiny portal veins. Let me get a different color. So you have all sorts of portal veins throughout the liver that are delivering blood. And that blood goes from a portal vein into a smaller venule, and at the each liver lobule is six-sided, and there are quite important structures at the corners, at the six corners. There are these triads. They're called portal triads. They're made up of a portal venule, which is smaller than a vein, a portal arteriole, and a bile duct. Those three structures are found at each corner. The portal venule is shown in blue. The portal arteriole is shown in red, and the bile duct is shown in sort of a yellowish green. Okay, the bile duct is here, same thing up here. So there are these triads at each corner. What happens is that blood draining from all of the, not just the intestines actually, but all of the organs in the abdominal cavity will enter into the liver through the hepatic portal vein, then go into it branches into many portal veins, and those branch into portal venules, and there's a portal venule, and here you see a branch going up into this triad and another one going into that triad. And so you have blood, venous blood that's nutrient rich, may have a few bacteria, may have a few toxins in it, that's entering into the liver lobule at the corners at the portal triad, and the blood then flows, and we should look here in this diagram at D here in the lower right. The blood then enters, it leaves the portal venule and goes in between these cords of, these are called cords, these rows of hepatocytes are called cords. You can see a row of hepatocytes here, there's another row of there, there's another row there, here's a row of hepatocytes. Let me get rid of that, what I just drew. Okay, so the hepatocytes are arranged in cords, and on one side, the venous blood coming from the portal venule flows, and it flows in towards the central vein. So there's blood coming in from the portal venules and going in, and as it goes in, it runs alongside the hepatocytes towards the central vein. And the hepatocytes process that blood. And among the many things they do through processing blood is they also produce at the same time bile. But you do not want bile mixing with the blood. And so the bile is produced between cords and flows back. It goes into this, it's secreted into this space here and goes back to the bile duct at the corner of each triad. So you have, I'll put it in a, I'm going to put it in a purple, and I'll do it with a pen. So you have bile being produced and flowing and being produced in the space in between the cords. So you have it here, that's the clearest spot, flowing back towards this. I don't like that color. You have it flowing back in this direction to the bile duct. And each of the bile ducts at the corner of each of the, within the triads, at the corner of each of the 6 corners of lobules, all will flow together to form, you know, the large hepatic ducts that drain bile out down to the gallbladder where it gets stored, and later through bile duct out into the duodenum when needed. In this way, blood is kept separate from bile that's produced. Now, the tissue of the liver, the hepatocytes that make up the liver, they also need oxygen, and they're not going to get oxygen from venous blood that's coming back from the intestines or other organs in the abdominal cavity and first flowing through the liver before it goes to get processed before it goes to the heart. They're going to get oxygen because there's also arteries that feed oxygenated blood into the liver, and that oxygenated blood comes in into the liver through the hepatic artery and goes into portal arterioles, and also flows in, flowing towards, mixing with and flowing towards the central vein, mixing with venous blood coming from the portal venule. So it all goes down, and in this way the hepatocytes on one side are exposed to blood, and on the other side they are secreting, and the blood is coming from either the portal venule and bringing in nutrient-rich venous return from the intestines, or bringing in blood from the portal arterioles, and it's oxygen-rich, so they get an oxygen supply, and it all flows towards the central vein. Meanwhile, bile is being produced and on the opposite side of the cord and flowing towards the triad portal, the triad, the bile duct in the triad, each triad. In this way, the organ gets oxygen-rich blood and nutrient-rich blood for processing and produces bile that's kept separate from that blood. Okay, what is bile, in fact? It's first of all, an alkaline solution. It's typically yellow or green or somewhere in between. It contains quite a few things. I would say the most important is that it contains bile salts. Okay, and it contains a number of other components that are listed here, but one of the most important things, the thing we'll talk about most, are bile salts. The bile salts are derived from cholesterol, and what they do is they help to emulsify fat. Emulsification means that the fat is processed in such a way that you can get tiny little drops of fat. You know, usually, like vegetable oil, for example, is a lipid like fat, and if you pour it on water, it'll all form one big glob, like it'll all coalesce together. And if you shake it up, you'll get little droplets of fat in the water, but when you stop shaking, they all coalesce together. And what bile salts do is they help emulsify. They'll help it to be present in small drops that won't come together. And what that does is it helps with fat and cholesterol breakdown and absorption. It helps to essentially kind of solubilize the cholesterol. It helps to allows you to have small droplets of fat. remember, the bile salts are in the bile, and the bile is being secreted into the small intestine, the duodenum. And that's where they act on fat in the food that you've eaten and allow it to get into small droplets. Otherwise, they'd all, join up together. They'd coalesce just like vegetable or any oil does when you pour it on top of water. You can watch it come together. Bile salts, by the way, they are secreted into the duodenum, will get recycled. They'll get taken up and recycled. The gallbladder is a thin green sac on the lower ventral surface of the liver. It stores and concentrates bile. There's absorption of water and some ions from the bile so that it's more concentrated. And it will release the bile down a duct into the bile duct and into the duodenum when a person eats a fatty meal. If there's more fat in the meal, you'll start releasing bile from the storage site of the gallbladder. And here you see that gallbladder, and it's actually has a muscular wall which can contract and squeeze its contents down into the space of the duodenum. Okay, so it will squeeze it down and release it here into the space of the duodenum if you have fat in the material that you've eaten. Okay, is there regulation of bile? Yes, all these functions in each system they're all tightly regulated, and in fact, if you want to understand physiology, it's regulation that's the key to understanding how systems operate. If you have entry into the duodenum of chyme, which is always acidic, it says acidic, but yeah, it's always acidic coming from the stomach. But if it's also fatty, okay, so you have entry of acidic and fatty chyme, that will trigger release from the duodenum, release from the enteroendocrine cells in the wall of the glands in the duodenum. You'll get enteroendocrine cells producing and releasing cholecystokinin, a hormone, CCK, and secretin. And of course, hormones always get secreted into the bloodstream. So the endocrine cells, it's not a large gland, it's just a few cells in each of the ducts, produce these two hormones. The secretin is transported through the blood and will go to the liver, among other places, but in the liver it is able to stimulate liver synthesis or production of bile. So it upregulates bile production. And here you see a acid fatty chyme going into the duodenum, triggering release of the two hormones, cholecystokinin and secretin. And they go into the bloodstream and they float around, and here you're seeing it as these red and blue dots, where the blue dots are secretin and the red dots are cholecystokinin. And the blue dots, the secretin, gets transported around and goes to the liver. Let me highlight that in a different color. Into the secretin stimulates the liver to produce bile more rapidly. So it upregulates synthesis of bile. Meanwhile, the cholecystokinin that was also released in response to acidic fatty chyme goes also through the bloodstream, and it does something different. It triggers contraction of the gallbladder and relaxation of the sphincters that control flow of bile through the bile duct, and allowing more entry of bile into the duodenum. Okay? So secretin upregulates bile production in the liver, and cholecystokinin triggers contraction of the gallbladder and relaxation of the sphincter that controls entry of the gallbladder of the bile into the duodenum. And that's what it says here. CCK will trigger gallbladder contraction and hepatopancreatic sphincter relaxation. And so bile flows into the duodenum, all triggered by the presence of acidic fatty is the key word, chyme in the duodenum. Meanwhile, the pancreas, which is also an extremely important accessory digestive organ, part of the digestive system, it's an accessory organ, just like the liver is an accessory organ. The pancreas has an important role. It has two types of tissue in it, exocrine and endocrine. So it is both an exocrine gland, secreting its products or secretions out of the body through the duct that leads down to the duodenum. So it secretes that enzymes, which collectively are called pancreatic juice, into the duodenum. That's the exocrine part and the exocrine function. And it also has an endocrine function, and has separate endocrine cells, and endocrine tissue or endocrine organs produce hormones, and hormones are always released into the bloodstream. The hormones that the pancreas produces, we've talked a little bit about them before, are insulin and glucagon, and they are involved in regulation of carbohydrate levels in the bloodstream and metabolism of carbohydrates. So these two hormones are produced by the endocrine tissue that's present inside the pancreas, and enzymes, hydrolytic enzymes or digestive enzymes, although I like the term hydrolytic better than all digestive enzymes are hydrolytic. So the hydrolytic enzymes, and there are quite a number of them, produced in the pancreas, they get excreted down through duct into the duodenum. And so you should consider the pancreas as being both an exocrine and an endocrine organ. So it has two functions. Here you see the structure in a drawing at a high magnification of the structure, the cellular arrangement inside a pancreas. There are tens of thousands of these inside a pancreas that is responsible for production of the exocrine. This is the exocrine tissue. This is the place where exocrine secretions are produced, the digestive enzymes. So you have, of course, small little ducts that lead to larger and larger ducts, and you have cells that are arranged in what's called a acinus. So you have these sort of sphere shape. Remember, we're just cutting, this is a cross section, so the sphere looks like a circle, but it's really a three-dimensional sphere, not a circle, but a sphere, okay, like a marble with a duct in the middle. And these acinar cells, pancreatic acinar cells, are producing the enzymes, and the digestive enzymes are always produced in inactive form. They're not active, they're in an inactive form. And inactive enzymes, are a general term for that is called zymogen. So these zymogens are produced and secreted here into this space of this duct, and the duct, here's another duct coming down, and they all go and fuse up with many other ducts that all together fuse into a large pancreatic duct that excretes down towards the duodenum. The pancreatic juice, as I said, is most of it is enzymes. There are also electrolytes, primarily bicarbonate. HCO3- is bicarb. It's quite alkaline. This helps to neutralize the acidic chyme and provide optimal conditions for the action of the pancreatic enzymes. Remember, enzymes have optimal pHs that they act on, at which they can be active. The enzymes, as I mentioned, are released as zymogens and have to be activated once they get into the duodenum. You don't want them activated before that, otherwise they would break down the actual tissue of the pancreas. That's bad. Okay, some examples of pancreatic enzymes that are produced as zymogens include trypsinogen, which once it's in the space of the duodenum gets activated to the enzyme, the active enzyme trypsin. There's procarboxypeptidase, which when it's activated, it becomes carboxypeptidase. Many enzymes that are inactive end in when you see this O-gen on the end of it, that means it's inactive. Other enzymes are named differently. Sometimes you'll see it with a pro in front of it. That again also means that it's inactive. So procarboxypeptidase is an enzyme, but it's inactive. It's not activated until it's within the site where it will carry out its enzymatic catalytic function. The enzymes that are secreted, there's an important one that's missing from this list. I'm going to have to add it here. There are not only is there pancreatic amylase and also lipases. Pancreatic amylase is similar to salivary amylase in that it acts on carbohydrates, particularly starch, to break apart and release monosaccharides from the polysaccharide starch. There are also lipases that help break down lipid and nucleases that help break down nucleic acids. But there are also, and what was missing from this list, there are also quite a few different enzymes that can act on proteins, and the collective term for that are proteases. And in fact, there's a couple of examples right up top, because trypsin is in fact a protease. It's an enzyme that acts on proteins, and carboxypeptidase also is an enzyme that can act on proteins. Another enzyme produced off the top of my head would be chymotrypsin. But anyhow, there are a number of different enzymes released by the pancreas. Enzymes that can act on proteins, proteases, enzymes that can act on nucleic acids called nucleases, enzymes that can act on lipid, lipases, and an enzyme one that can act on carbohydrates, which is amylase. Okay. Similar to bile production and release, which is affected by secretin and cystokinin, cholecystokinin, when we talked about the liver, right? Pancreatic secretion of enzymes is also controlled by these same enzymes, hormones rather, secretin and cholecystokinin. If a person eats fatty food, or there is acidic chyme entering into the duodenum, remember that's an important point, or. With bile, it was fatty, acidic and fatty. Here it's either fatty or acidic, it doesn't matter. If the chyme is there, it's either acidic or fatty or acidically fatty. You know, if it's both, that's fine too. It will trigger cholecystokinin and secretin release, and that acts on the pancreas. So those will go to the pancreas and act on the pancreas, inducing secretion. Cholecystokinin, in this case, induces secretion of the pancreatic juice from the pancreas, and secretin causes secretion of a bicarbonate-rich pancreatic juice. So that bicarbonate-rich pancreatic juice can help reduce the acidity of the chyme. So here we have enzyme-rich, and here we have bicarbonate-rich, cholecystokinin triggering enzyme-rich pancreatic juice, and secretin triggering bicarbonate-rich pancreatic juice. And again, a similar diagram to the one we looked at before, where acidic or fatty chyme enters the duodenum, causing enteroendocrine glandular cells in the wall of the duodenum to release secretin. Fatty, protein-rich chyme will induce release of cholecystokinin. The cholecystokinin and the secretin enter into these two hormones enter into the bloodstream. When they reach the pancreas, the cholecystokinin induces secretion of enzyme-rich pancreatic juice, and secretin will trigger release of bicarbonate-rich pancreatic juice. Remember, bicarb has a high pH. It's alkaline. It can neutralize the acidity of acid chyme. That's the end of Unit 8, Part 3.